Supérieure and Sorbonne University to give us the next talk. Right. You can get started now, buddy.
Okay. Okay. So hello, everybody. No, I go back here. So I hope I will have no problem for my presentation for John that I know from a long time ago. And because I am in Italy at the moment in a hotel and the connection is bad. So if it stops, it's not my fault. Okay, so the work I will present today is something we began some time ago with Institut Curie, who is a famous institute for cancer research and who has a very good hospital, which is close to my lab, very close to my lab. And we focus about immunology and cancer. And what I present is simply the interplay between T cell, fibroblast, and cancer cell. It's limited to that because we have data for that. Okay. So I think, okay, for you who are specialists of cancer and especially Jean, perhaps it is appropriate for him. I don't know. Okay, so I go. So as I tell you, since 4 years, we have this collaboration with the people of Curie and especially the people concerned by the lung tumors. And it's the biologists are in a immunology lab. And so my presentation concerned primary solid tumor of the lung. And I immediately give you all the cell I consider, because there is plenty of cell, of course, each time, plenty of bad cell each time. And so the cell can be macrophages, but not in this talk. T cell and B cell, which are cells of the immune system, but for this talk it will be T cell mostly. And of course, cancer cell and eventually, and also healthy cell. And also you have all the chemical signaling. So we have a special focus of a lung carcinoma, which is called, you have this name, so I do not remember, but the first letters are for non-small cancerous lung carcinoma, I think. And in this case, what we need is we can restrict on fibroblast at least for the moment. This is the only things we can do because we do not have data for macrophages, T cells, and cancer cells. So this work has been done mostly by a PhD student, now who is a postdoc in a NEBO lab, Joseph Ackerman, and with a group of Napoli in Italy, Chiara Bernard and Massimiliano Fraldi, and for the biologist Philemon Sirven and Hélène Salmon. So, as I say, I focused on the most active cell for this problem. And so what we need to sum up the things, you have cancer cell, which proliferates, which are eliminated by the T cell, at least is the hope, that the T cell succeed to eliminate the cancer cell. But you have also in the stroma fibroblasts which are here because you know in stroma you have fibroblasts, and because we are close to a tumor, they are transformed into what we call activated fibroblasts. It means that they change their property in proliferation, and of course property concerning the T cell, and they are called also CAF usually. And the T cell, of course, are attracted by to the cancer cell, but they are inhibited in several ways, chemically, and they can stop in the bad cases by the fibroblast. So this is the things we have to treat together, to couple together, and of course the chemical signaling. So there is a typical picture in immunology where it's possible to classify the tumor, the solid tumor, and into immune desert. Immune desert means that our immune system is completely inefficient. Immune excluded, so it means that the immune system reacts to the presence of a tumor and try to do the job he has to do, but is excluded from the interior of the tumor, and hot tumors, which is the beta case, because you have T cell inside the tumor and some hope for treatment. And this picture is also very important for the immunology treatment, in the sense that somebody who has an immune inflamed or tumor, will be more sensitive to the CAR T, for example, immunology. Okay. So if we consider all the cells, we have to consider, and we write simply a dynamical system, and I think it's important to begin with a dynamical system in order to see the parameters which are important for the following. If you consider these parameters, you begin by an ordinary nonlinear equation for the cancer cell, for example, very simple, and you say, okay, the cancer cell, their role is to proliferate in the first time, and after they saturate for another reason, is that the quantity I consider is the density. So it means I consider not the number of cancer cells, but the ratio between the volume of cancer cell and the total volume of my space. And of course, this ratio is smaller than 1. So we have to say there is a natural saturation that you can understand as a pressure eventually if you want. So you have a parameter which is delta in the first equation. In this equation, I don't know if you see when I do that. The equation.
Yeah.
Yes, you see. Okay. Immediately there is one parameter, and you can say, okay, I limit the number of parameters, so I can choose my unit of time, which is the proliferation rate for the new volume, which is not exactly the proliferation rate of the cancer cell, hopefully for us, but it's something, but it's a unit of time that you can choose. So there is one parameter which is eliminated, which is present but eliminated because it's our scale of time. And this is quite the only one for them. And after you have the T cell, and this is what I say, the T cell are attracted, the second equation, which is here, are attracted by the cancer cell. Because they are made in, they originate from the thymus, I don't know, the thymus, and the thymus is not at the level of the lung, it's something elsewhere. And so they are attracted because there is an inflammation place, situation. So they are attracted by the cancer cell. And also, they disappear, they are limited naturally, but now the pressure is made with the cancer and the T cell. And if you look to the first equation, now the cancer cells are attacked, in the good case, by the T cell. So now you have 3 coefficients, only that, only for that. And 2 coefficients, which will be rather important for the situation, which is this one and this one. Huh? So there's 2 delta, if you want. There's 2 delta are especially important in the following. And you have what happens with the fibroblast. So, sorry, what I have done. Je veux remonter. Ah, sorry, I am very sorry. So, the fibroblast. So the fibroblast, as we said before, the fibroblast now they make 2 families of fibroblasts. The fibroblasts which are in situ, if you want. And this is the non-activated fibroblasts, so they are here. A priori, their number increase because of the cancer cell, because cancer cell may transform cells. So they are transformed by the cancer cell. They disappear because they are transformed into CAF, or associated fibroblasts. And after you have the same quantity, the same delta. But this function has become more important. So as soon as you introduce new participants, you have more coefficient. And when we study all the coefficients, we arrive to a number which is 11. And 11, it means that you can do what you want. You obtain what you want with 11 coefficients when they are not known. But in fact, we realize that there are 2 important coefficients, which is the delta, which represents the pressure, which limits the growth of all these quantities, and epsilon, which is the ratio between delta and the coefficient of the death of the cancer cell due to the T cell only. And depending epsilon, this epsilon and delta, in fact, you succeed to represent all the scenario for immunology that I have represented at the beginning. And in particular, we analyze a little bit. We were happy to have this picture of tumor of the lung given by Institut Curie. So it's the case of 2 patients. And if you look at the tumor sites are in gray for the 2 patients. So it's very far from the disc in 2D of a growing tumor, as you see, because the tumors are a little bit distorted. So but it is like this. It's bad news for us. And if you look now, where are the T cells for this patient? They are here, so they are not in the tumor. So this patient has attract, obviously, T cell, but they do not penetrate the tumor. And here they put ANOVA signaling, in order to visualize the fibroblasts. And the fibroblasts are in red, and the fibroblasts are, as I mentioned before, mostly located outside the tumor, not inside. And I don't know if they do a protection of the tumor, but probably this is their role, and it is a bad role for the patient. And also you have over cell, but a small number of cells. And if you look at the second patient, which is below, you see that in the tumor, which is here, the tumor is here. Hopefully we have a drawing to understand what happens, because it's rather, so for people who are not specialists of figures, it's difficult. But you see, you have a black zone inside the tumor. And I ask if they can tell me if it is the necrosis, the tumor necrosis or not. And they say because may take caution, and they say perhaps, but it's probably because it is the place where you have plenty of T cell inside the tumor, yeah, and plenty of T cell. Here again are the fibroblasts. So you have two patients which are in two different situations, which correspond mostly to the scheme of the beginning. At least you have a reaction of the immune system, but which is not enough. So now it is true that what it is important is to treat the space, because it's our system as, okay, we analyze the system, but for many people now, they may think that it is okay, we do better now for the system, but we want something simple. And now we have the space equation. So this is really inspired also from, I want to say, from the paper by John and the review paper. He has written, 15 years ago, about modeling the model of tumor. And so the first equation is the concentration of cells I, so cancer cell, fibroblasts, and so on. And here, the right-hand side of this first equation is the equation, the coefficient exactly of the dynamical system. So it's easy, but it is written, it's a linear system, so it's easy to write too. And you have such equation for the growth of a species, for example, cancer cell, but a velocity. And for all the neutrons, chemical signaling, and so on, it's easy because it's a diffusion equation. It's a diffusion equation with a term for which express attraction of this chemical by a species, and the depth, the uptake. And but the main problem is how to find the velocity, and the velocity itself. So it's more. This is the difficult point: is to find the velocity of the species. And for that, we use the relation method equivalent to the other dynamical equation. I'm sorry, very sorry. Okay. And so the relation means simply that the velocity we must make extremal the dissipation. So the dissipation to calculate, so it's allowed to obtain a very more easily, I will say, the velocity. In fact, I prefer this method. This method is due to Lord Rayleigh. His name was Strutt when he was not a lord, but after he becomes Lord Rayleigh. And this is a very old publication. But anyway, if you use the relation, like we use the Hamilton equation and so on, it simplifies a little bit the formalism. And formalism, I say it is safe. It means that you know where to put the phi. And okay, and after you have the dissipation, so, and we take a fluid a fluid friction, either Stokes, either Darcy. Darcy is probably more appropriate, but okay, you have both possibility. And we add an energy, interfacial energy between phases. So it remains the interaction between cells. In our case, we have chosen the Fleury energy, which takes into account the diffusion of the coefficient of a species and the interaction between cell-cell interaction, more specifically, and to limit also heavy calculation, we take interaction between cancer cell because, of course, cancer cell attract each other since we make an aggregate or a tumor. I have forgotten to say that in the lung, you have really nest. So it means high concentration of cancer cell localized in a space. Localized in a space. It's really nest. So with a percentage around 90%. So it's a huge percentage. And the T cell attracted by the C, this is what I say, this coefficient. And finally, the fibroblast and the cancer cell. So this is all attractive term and only limited to the most active cell. Dissipation is ordinary. It means Stokes flow for that or Darcy flow for that with the concentration here. So now I will give you simply, I will give example of simulation. So as you have seen in the first photo, in the first slide, the two nests can be rather close to each other. And we ask the following question: it's a simple geometry if we can have a collapse of the tumor. The two tumor seeds can be joined together to make a big tumor. And of course, it depends on our parameters. It's not always true sometimes. And it depends if we have a good inflammation of the tumor. In this case, the nest disappears before the collapse. Or if you have an intermediate situation, and because we have a strange geometry for the tumor, we try to mimic the geometry, but there is work to continue on that because we have this paper is under review, and it's one of the questions of the referee, why we limit ourselves to circular tumor, and we are not, okay, circular, of course. So I continue. I have another slide before this, but obviously, voilà. So, so to sum up, so I think for this work, okay, we have to improve the representation of tortuous tumor, and we will succeed for sure. Now, the next step is, of course, to understand the macrophages. And you know, the macrophages, they are like their role, you know, you have always macrophages in your tissue. It's like the policeman in the city, you know, you have always, and they are ready to attack eventually. And to see, anyway, we have information, in vitro information about how they are attracted by the cells and so on. And this is our next step. And finally, this is our paper concerning that. What I want to say is that my presentation is quite accepted in eLife at the moment. So finally, I would like to say to Jean, happy birthday. Unfortunately, I cannot be in Canada because I have another celebration in Italy. And Italy is a little bit less expensive and complicated to go. But okay, I would like to see him really, and I hope to see him in the next future. Thank you very much for your attention.
Thank you.